Skin is the human body's largest organ, weighing some 8 pounds and stretching almost 22 square feet. With skin cancers forming the most common groups of cancers diagnosed worldwide, there is a growing urgency for more insights that could lead to new diagnostics and treatment approaches. Yet among all cancers, there is relatively little understanding of their pathogenesis and how they develop. Being able to study the space-time relationship between cells is changing the game in cancer research. Exactly what Professor Marine's work is focused on.
Melanoma is a very aggressive form of skin cancer, and we believe that it is in part due to the fact that it's a very heterogeneous disease. This disease is composed of many different types of cells. We still don't really understand what these cells do in the tumors and how they contribute to the spreading of the disease. So genomic analysis is a method to analyze the genome of every cell. In a way, is to build an identity card of every single cell. And so we are able to today, with these technologies, to discriminate between different cells in the tumors and thereby understand heterogeneity.
Stereo-sequencing is a method developed by BGI Group that allows us to map the cells in the tissue. With that technique, we can identify every single cell and position these cells in the tissue. That was not possible in the past. It really opens up new possibilities to understand cell-cell communication.
So we've used Stereo-seq and a number of other technologies to dissect the intratumor heterogeneity that one can find in melanoma. This helped us to identify different subpopulations of cells, some that fuel growth, that contribute to the expansion of these tumors, and other subpopulations that fuel metastasis, meaning dissemination or spreading of the disease to different organs. These two subtypes are important because one fuel growth, so by eradicating that population, we might be able to decrease tumor burden and tumor growth. And most importantly, by understanding and potentially eradicating these cells that disseminate and spread to different organs, those metastatic initiating cells, we might be able to block the spreading of the disease and thereby the dangerous phase of the disease. But this is a paradigm shift in the field because we show that this cancer stem cell phenotype can be acquired through reprogramming. This is very important because it shows that tumor growth can be stimulated by almost any cells in the tumor if they are in close contact to blood vessels. Understanding that, of course, allows us to potentially develop therapeutics that target this communication between endothelial cells and cancer cells and thereby block tumor growth.
Working with researchers like Professor Chris Marine and oncology experts like Professor Bechter is what drives BGI Group to develop more innovative ways to map the world's DNA to help us better understand the basic building blocks of life.
What we have learned in the past years is that the complexity of the tumor is so big that you need to focus down, you need to boil down to really cell-cell interactions. And you need to understand which cells communicate with each other. And only if you decipher this communication pathway, you will be able to intervene with a treatment strategy. The more and the better we understand the heterogeneity within a tumor, the more we will be able to delineate new treatment strategies which actually aim at the heterogeneity which is present in a given tumor. So we will be able to, tailor treatment strategies more based on the knowledge we have from genomics.
So using the BGI technology, we've been able to identify and map important subpopulations of melanoma cells in the tissue. These technologies really teach us a lot about how these tumors grow, how these tumors spread, and I think we've learned quite a big deal already, but this is just the beginning. These technologies will really allow us to better understand these processes and eventually how to interact and intervene therapeutically.